Hey everybody, welcome back. This is the first of a two-part lecture on our sensation and perception unit. This lecture will focus specifically on our senses, and our next lecture will focus on our perceptions and how we make sense of the different sensory experiences that we have. So we're going to start with our sense of vision. Vision is certainly the most detailed sense that we go into in AP psychology, and there's a lot with our sense of vision. That said, a lot of times when we are assessed on vision, it's in the multiple choice section with different questions trying to figure out if we remember the anatomy of the eye and things like that. Vision does not show up very much in the FRQ. It's more likely that we would see questions about perception on the FRQ than the sense of vision. But I'll point out a few things that we've seen in the past. So I have at the top of this slide the anatomy of the eye. It's kind of a simplified version of what's happening. But recognize that as light travels through the eye, the first structure that it's going to kind of run into is going to be your cornea. And your cornea is the outermost layer. It helps protect the eye and it helps bend light toward the center of our eye, which is where the pupil is. And again, the pupil is that small black hole in our eyes. And it's surrounded by this muscle that we call the iris, and it's the colorful part of your eye. When you are in a situation with a lot of really bright light, then the iris is going to constrict your pupil and make it small. Whereas if you walk into, let's say, a movie theater or someplace where there's not a lot of light, then your iris will dilate your pupil, make your pupil very large to allow more light to come in. The light is then going to be focused with the lens, just like a camera lens, and it'll focus that light onto the back of your eye. The lens is concaved, and as a result of that, the image itself flips upside down. So you can see in this example here, there's a candle flame, and by the time that image reaches the back of the eye, the candle is inverted. Most of that image is going to be centered on the focal point, what we call the fovea. And for your foveal vision, that was an FRQ term a few years ago, foveal vision. You could look at a piece of paper or a book or something, and you can find one word, and that one word is probably pretty crisp. It's in detail, and you can see it clearly. And then while focusing on that one word, you'll notice that the further away you get from that, the word above and below to the sides, they are less in focus. And it's because that part of the image is being shown and projected on different parts of the retina outside of the fovea. The next slide will go into a little bit more detail about the retina and what's contained in the retina with these different photoreceptors. And then obviously that message is going to be sent down through your optic nerve. I have some noteworthy vocab here. Transduction is not unique to vision. Transduction is the process of converting one particular energy or stimuli into an electric message that your brain can interpret. So your brain doesn't make sense of light waves. So instead, we have transduction in the retina, which converts light waves into an electric message that you can understand. Same with sound waves. Your brain doesn't know what to do with vibrations in the air. So what happens is you have transduction in the ear, which converts sound waves into an electric message that your brain can interpret. Parallel processing is this idea that you can interpret lots of different aspects of vision all simultaneously. So I give the example of a bird flying. If you're outside and you see a bird flying in the air, you can see its color, its shape, its movement, and you can see all of those things happening simultaneously at the same time. That's the idea of parallel processing. And then feature detectors are neurons. They're interneurons that are found along the visual cortex on our occipital lobe. And feature detectors are neurons that respond to very specific angles and shapes and things. And so if you're looking at an object that has a vertical line in it, there are certain neurons that will respond very heavily to that particular stimuli, whereas other neurons or other feature detectors in your brain respond to horizontal lines. And so we can, through brain mapping, we can track what types of objects you're looking at by seeing which different neurons are lighting up while you're looking at different things, which is pretty cool. So as I said, a little bit more specific of the retina itself, there are different types of neurons and cells that exist within the retina. perhaps most important for this class is understanding the difference between rods and cones. These are photoreceptors, or they're cells that are responsive to light. And rods are very light sensitive. They're found along your peripheral vision, and they help us see at night. They help us see in black and white. Whereas cones, you can think the CO in cone helps us see in CO in color. Cones are located along the back center of the eye, and it takes a lot of light in order for cones to work. There's one theory as to how we see different types of colors, what we call the Young-Helmholtz trichromatic theory. And the idea behind this theory is that you actually have different types of cones. You have red cones, blue cones, and green cones. And combinations of those different red, blue, and green cones allow us to see all the different colors of the rainbow. Another theory of color vision is what we call the opponent process theory. And this one's a little bit trickier to kind of wrap your mind around, but the idea is you have to think that different cells are paired together. And so we have red with green, you can think Christmas. We have blue and yellow, and then we have black and white. And if one of those channels is being overworked, right? And so if you're looking at something that's really red, and so I've got the red part of my Chiefs hat, and if you're looking at something that's really red for a long period of time, well, green is not able to go down that same channel. So then when we take red away, it produces the perception of seeing green, what we call an after image. And so after images provide some evidence that opponent processing theory works. Don't think that these theories are one or the other. They both help us really understand how we perceive color. For our sense of hearing, there are a few structures that I want to draw your attention to within the ear. One is the cochlea, and the cochlea is kind of this snail-like structure. And the idea here is that the cochlea is part of our inner ear. As sound waves travel down through our auditory canal, hit our eardrum, the eardrum vibrates with that sound. There are these small bones that exist in our middle ear that move with this vibration, and then it pushes this sound into our cochlea. The cochlea is lined with what's known as the basilar membrane. And the basilar membrane was an FRQ term last year. Lots of students confused that with the basal ganglia in the brain. But the basilar membrane has these small hair cells, and they're neurons, and what they do is they respond to vibrations in the air. And so that's where the transduction takes place. Again, your brain doesn't know what to do with vibrations, but it certainly does know what to do with this electric message that's being produced by the basilar membrane. The only other structure I really want to point out to you actually doesn't have much to do with hearing at all, but these are these three tubes that you have in each ear, what we call our semicircular canals. And in those tubes, there's liquid. And what happens is that as that liquid moves around, there are these sensors, these sensory cells on the inside of your semicircular canals that help detect movement. So your semicircular canals helps send messages to your mind about if you're standing upright, if you're lying down, if you're spinning really quickly in circles, if you're on a roller coaster. As that liquid in the semicircular canal sloshes around, it's giving insight to your mind as far as your body's position in relation to gravity. We perceive sound in a couple of different ways. When we think about an actual sound wave, the pitch of a sound, think high or low, is going to be decided by the wavelength itself. And so if we have really long, drawn out sound waves, that's going to produce a really low pitch. Whereas if we have short sound waves that are very closely crunched together, that's a really high pitch that you might expect from like a violin or a flute, something like that. Amplitude looks at the height of a sound wave. And so with sound waves that have a really tall amplitude from what's called the crest to the trough, those produce really loud sounds, whereas short amplitudes would be quiet sounds like a whisper. As far as pitch itself, if we go into more specifics into detecting things that are high-pitched or low-pitched, some different theories that we have there. Place theory is simply the idea that along the cochlea, different neurons will respond to different pitches. And so if you have, I'll back this slide up just a little bit. This is our cochlea right here. If you have sounds that are registering toward the base of the cochlea, those are going to be our really high-pitched sounds. whereas sounds that are registering really low toward the end of our cochlea, those are going to be our lower pitched sounds. So that's the idea of place theory. It's simply the location on the cochlea itself. Frequency theory, on the other hand, is the idea that neurons will fire essentially with the frequency of a sound wave. And so as that sound wave is traveling through the air, think for every sound wave we have, that neuron is firing. But neurons can only fire so fast. And so because neurons can only fire so fast, we have to bring in this idea of what's called the volley principle. So maybe you hear a sound that's 20,000 hertz. Well, your neurons can't fire that quickly. And so because your neurons can't fire that quickly, we have a cluster of neurons surrounding neurons, and the sum of their firing all together can equate to that really high-pitched sound. And then I have down there at the bottom some different types of hearing loss, both conduction and sensory neural. Conduction hearing loss, that's usually we're talking about things like rupturing an eardrum, which might happen if you go up into an airplane too fast, or if you dive really deep into a swimming pool, you might have some conductive hearing loss, and that's usually repairable. Sensory neural hearing loss, on the other hand, that usually takes place in the cochlea and happens not usually because of like one particular event, but exposure to loud sounds or exposure to high-pitched sounds for long periods of time. You can damage those neurons in your cochlea and can lead to sensory neural hearing loss. And the therapy for that is certainly not as advanced, and the hearing loss itself is certainly more long-lasting. Real quick, some of our other senses, our sense of touch, or our body senses, we have different neurons for pressure and warmth and pain and cold. We talked a little bit in class about the biopsychosocial influence of pain. Pain is a perception, which is a really weird thing to think about. But if you stub your toe, that experience of pain takes place in your mind. It actually doesn't take place in your toe, which is a weird kind of concept. And there's evidence for that with people who are paralyzed, right? If you have someone who's paralyzed from the waist down, if they were to somehow, have something drop on their foot, they wouldn't experience pain because that message isn't getting sent to the brain. Gate control theory operates under this idea that pain messages are sent up through your spinal cord, and your spinal cord has these gates that can open and close, and we can flood your spinal cord with other neural messages, such as the message of pressure. We've all had that experience of hitting our shin, and when that really hurts, we kind of squeeze our shin really hard, and that seems to help with the pain. Well, the reason that's helping with the pain is because we're flooding that spinal cord with the message of pressure instead of the message of pain. Olfaction and gustation. Olfaction is our sense of smell. Gustation is our sense of taste. Think your nose is like an old factory. Think that your sense of taste, if you're hungry, you might go to the gas station. And one thing that we see often with these terms in AP Psych is the idea of sensory interaction and how our senses work together to create a perception. We did the starburst activity where when you eat a starburst, it's not just your sense of taste, but it's also your sense of smell to give you the perception of flavor. And if we plug our nose, you don't get that sense of smell, and it hurts your ability to be able to detect flavor. And then the difference between kinesthesis and vestibular senses. Your kinesthetic sense is going to be your body's position in relation to itself. And so you have sensory neurons all over your muscles and your tendons, and it's your ability to take your hands and interlock your fingers behind your back. It's your kinesthetic sense, your body's position in relation to itself. It's why you can walk without having to watch your feet to make sure you stumble over your own feet. Your vestibular sense, on the other hand, those neurons are located in your semicircular canals, and your vestibular sense is your body's position in relation to the world, so that you know whether you're standing up or laying down or something like that. That's going to do it for our presentation on senses, and our next presentation will be again about perception. So I hope that's helpful. Good luck.